Greetings, ladies and gentlemen, or as I like to call you, biology students. Welcome to your meiosis notes, also known as meiosis. Please get out your fill-in-the-blank notes if you did not do them in class, or if you need to correct any blanks. Let me know how this goes after you've watched it. Next couple of slides were already taken care of in class, but I want to go over them in case you're doing this as a review. So as far as sets of chromosomes go, we did talk about the fact that haploid cells have one set of chromosomes, diploid cells have two sets of chromosomes. Human normal body cells have two sets of 23 chromosomes each. Now, each of those 23 chromosomes has a pair, a matching pair, or what we call a homologous chromosome that goes with it. What these chromosomes have in common is they contain genes for the same traits. You get one out of each pair of homologous chromosomes from your mother and one from your father. And while these have genes for the same traits, they can have different versions of that gene. For example, if you have a gene for eye color, you could get a version for the brown eye color from your father and the blue eye color from your mother. But they're still genes for the same trait, and they would be located on the same chromosome in the same location for each of those two chromosomes. We've already mentioned ploidy, that is, haploid and diploid cells. These are the only types of ploidy you'll need to know about for this course and for our test. So haploid cells, you can remember that by thinking hap, half, sound very similar, and they have half the normal amount of DNA that you would find in most of your cells. The only haploid cells that you find inside humans are sperm and eggs, otherwise known as the gametes or sex cells. And these guys only have one set of chromosomes. That's indicated by the 1N that you see in parentheses after haploid cells. N stands for the number of chromosomes in one set. Diploid cells make up most of our body. They contain two sets of chromosomes, and these are also known as somatic or body cells. Please keep track of the difference between haploid and diploid cells, because later on when we talk about the actual process of meiosis, we'll be keeping track of at which points in the process you have haploid and at which point you have diploid cells. This slide is just pointing out the fact that gametes are sex cells. So you have sperm and eggs that are gametes. And then once a sperm fertilizes an egg cell, that fertilized egg cell is called a zygote. Keep that in mind. We'll talk about it with genetics. We'll also do a review of specialization and differentiation in developing embryos, at which point you'll need to know what a zygote is. Wanted to revisit the idea of homologous chromosomes very quickly. So what you see here is a visual, an example of what you might see as a maternal copy or a copy from the mother, and the paternal copy or the copy from the father. I want to point out that in actual cells, your chromosomes are not color-coded by origin, whether they're from your mother or from your father, and they don't exactly look like what you're seeing here. This is kind of a very generalized image of a chromosome. Chromosomes do have these patterns of light and dark bands, and you can use these patterns of light and dark bands to try and match up which are homologous chromosomes. Notice the chromosomes are the same length, and also the centromeres are in the same position. When we go over this in class, we'll practice matching homologous chromosomes, so you'll be able to figure out how to do this. Concept that we'll be revisiting later is that of what's called a karyotype. I want to remind you when we talked about eukaryotic and prokaryotic cells, I mentioned that karyo means nucleus, so literally karyotype means nucleus type. Basically what this happens in doctor's offices or with geneticists, they'll take a cell, they'll split up, they'll lyse it, and they'll pop open the nucleus, and they will take images of each of the chromosomes. When they do this, they'll match up pairs of chromosomes, the homologous chromosomes, and they use this basically to look for abnormal numbers of chromosomes. You may, in fact, want to go ask your parents if you have a karyotype, or basically an image of your chromosomes. It's not done all the time, but you may just have one. Example, this is the karyotype of the son of one of the teachers who used to work at Sunset several years ago. So this was an actual karyotype done. Notice the chromosomes, while they kind of look they don't look exactly neat and nice and easy to look at, you can pretty much match up these chromosomes based on the pattern of the bands, their length, and a couple of other characteristics. We're going to look at karyotypes. We'll look at chromosomes and such things in karyotypes a little later when we talk about genetic disorders in more detail. Now, one of the things that we're going to talk about quite often, and it's going to be a recurring theme, is the fact that meiosis and mitosis have a lot in common. One of the things that they have in common is before both processes, you do actually undergo S phase, which means that you, since S stands for synthesize, you synthesize your DNA, make a new copy. So to make a little bit of sense about what's going to come out at the end of meiosis, I want to point out how many sets of chromosomes you find in a cell after S phase, because this is what a cell is going to look like, or this is what it's going to look like right before meiosis. So we're going to count that in the next few seconds. Most important and probably most confusing thing to keep in mind is that, as you see here, you have a pair of homologous chromosomes. The purple ones come from the mother and the blue ones come from the father. But it gets more complicated because each homologous chromosome is made of a pair of sister chromatids. So hold on, and we're going to count the sets of chromosomes. So if you wanted to get technical about it, you could technically say that each chromatid is its own set of chromosomes. So here we have set 1. Notice we have another set of chromosomes in the sister chromatid of that original chromosome we're looking at. We look at the homologous chromosome, the version that came from dad, we see there's the first sister chromatid makes up the third set of chromosomes in the cell. And with the sister chromatid of that homologous chromosome, the one that came from dad, you have your fourth set of chromosomes. Don't worry if this confuses you. We'll go over this more in class, and if you have any questions about this, you can make a note of it. I left a special section on your written notes so that you can write down any questions you may have, either while watching this set of notes or while going over it in class. So, as we go through the steps of meiosis and the process, please keep in mind the difference between homologous chromosomes. They are not clones of each other, they are not identical copies, and the difference between sister chromatids, which are exact copies of each other. Remember, homologous chromosomes, one came from mom, one came from dad. With each pair of sister chromatids, you're looking at, say, the chromosome you got from mom, and another pair of sister chromatids could be the chromosome you got from dad. So we're going to look at this. If you get confused, feel free to pause, rewind, rewatch, because we're going to use a lot of terminology that we're just starting to get familiar with. So one of the things that we'll end up doing in class is we're going to compare and contrast mitosis and meiosis in several ways. So just like in mitosis, right before the process happens, you do duplicate your chromosomes. But unlike in mitosis, you're actually going to have your homologous chromosomes pair up with each other. And then you're going to do the equivalent of performing mitosis 2 times in a row. This is going to be done without reproducing the chromosomes in between, so the end result will be that you cut the number of chromosomes you have in a cell in half. As if your lives weren't already complicated enough, there are 2 parts to meiosis, meiosis 1 and meiosis 2. They do different jobs, although they do have the same stages happening. So before we get into that, let's look at what happens in meiosis 1. Meiosis 1 is going to separate homologous chromosomes. That's going to end up giving you 2 cells which each have 23 pairs of sister chromatids. No homologous chromosomes in meiosis 1, at the end of meiosis 1. In meiosis 2, you're finally going to separate those sister chromatids, which is very similar to what happens in mitosis. And the end result of that is you're going to have 4 cells total now. Each of them is going to have 23 single chromosomes. Keep in mind this number 23 means we're looking at human cells, but as long as you know the number of chromosomes in a set, in any species, the same principle applies. Big reason why I tried to teach you guys this 1 or 2 word phrase to help you remember what happens during each phase of mitosis is that even in meiosis, meiosis 1 and meiosis 2, if the phase is named prophase, prophase is always preparing for something to happen. There are metaphases in meiosis, just like in mitosis, and just like in mitosis, you're going to have some chromosomes lined up at the middle of the cell. Anaphase is always pulling things apart, and telophase is always the end. You're wrapping things up and getting ready for the next stage in the cell cycle. We have a nice complicated diagram that shows you all the stages of meiosis 1. Don't worry if it looks kind of confusing. We're going to go through each of these stages one by one. You'll note that you do have a data table in your notes with blanks and everything in it. That goes with the next several slides, which will detail what happens in each stage of meiosis. Talk about what you're going to see on these slides. The center section of each slide is going to show an image, both a microscope image and a picture diagram. So on the left-hand side of the image, what you're going to see is a description of what you end up with at the end of that phase. So at the end of prophase 1, you still have one diploid cell. You haven't split the cell apart into two, so there's still one cell. It's still got pairs of homologous chromosomes in it, and each of those homologous chromosomes has an identical sister chromatid attached to it. So everything's still diploid at the end of prophase 1, and during prophase 1, your homologous chromosomes pair up with each other. They physically connect, and what we're going to look at on the next two slides is they perform something called crossing over, where they exchange sections of DNA. Now, I mentioned that during prophase 1, homologous chromosomes pair up with each other. When I say they pair up, they're actually going to join together at their centromere and form these structures called tetrads. While they're paired up, their chromosome arms, as we call the parts that come away from the centromere, can overlap each other. And where they overlap, they can actually swap out sections of chromosome. This is going to be really important when we start getting into more detail in genetics. And it's going to create not only some what we call genetic diversity, meaning different combinations of genes, but it's also an area where mutations or changes in DNA can happen. And don't worry, we're going to talk about mutations later, but we're not going to watch X-Men. When homologous chromosomes swap sections, this is called crossing over. And this is really important because it creates new combinations of genes. So if you'll look at or keep in mind the previous slide, you noticed you had an entirely purple chromosome and you had an entirely blue chromosome. The entirely purple chromosome came from mom, the entirely blue one came from dad. Well, after crossing over has happened, you will see now you have chromosomes that are part from mom, part from dad. So on the left on this screen, you see a chromosome that the most of it comes from mom, but a small chunk of it is your paternal chromosome, or the one that came from dad. So let's say that you had genes for a couple of different traits on these chromosomes. You may end up with a mix of mom and dad's traits on the same chromosome. We'll talk about this more with genetics. We'll talk about the importance of this, but since it happens at this point in meiosis, I wanted to make sure and point it out. So the stages of meiosis are going to happen in the same order that the stages of mitosis do. So after prophase is going to come metaphase. Now we're looking at metaphase 1, which is of course, metaphase of meiosis 1. And in meiosis 1, all we're doing is splitting up homologous chromosomes. So here we have these pairs of homologous chromosomes line up along the middle. I want to point out that on the left-hand side of the picture where I have written what happens at the end or what you get out of the end of this process, since it's the exact same as what happens at the end of prophase, you still have one cell, it's still diploid, I've basically grayed this out so it doesn't draw your attention. I want to also point out on the picture, if you'll look where the chromosomes that are in the box that has a sign pointing to it that says homologs align, notice how those chromosomes have parts of each other. They've exchanged pieces, so you see crossing over has occurred between those homologous chromosomes. Always in anaphase, you are pulling apart chromosomes. Here, you're separating homologous chromosomes from each other, pulling them to opposite ends of the cell. Notice there's something different at the end of telophase. Since you have cytokinesis happening at about the same time, you're going to end up with two haploid cells because you've separated homologous chromosomes. But each of those haploid cells In each of those haploid cells, every chromosome is paired up with its sister chromatid. That's the part that can get confusing. You've separated, so now in each new cell that's being formed, you do have two sets of chromosomes. They're just exactly identical to each other. They're copies, they're clones. So we count that as a haploid cell because you don't have homologous chromosomes anymore. We have an image that's an overview of meiosis 2. Don't worry if I flash past this image too quickly because you do have a copy of it in your notes. Alright, from this stage on, during all of meiosis 2, you'll see two cells in each image. Why? These are the two cells that were produced at the end of telophase 1. Now, I want to point out there is no new DNA made in between telophase 1 and prophase 2. Basically, most important thing, you're making a new protein spindle and you're attaching the pairs of sister chromatids to it. That's all. Metaphase 2, these pairs of sister chromatids line up along the center of the cell, they're in the middle, and you end up separating. You're going to separate them during the next stage. During anaphase 2, you separate those sister chromatids from each other, and they're going to move towards opposite ends of the cell. And finally, we end with telophase 2, where you have cytokinesis splitting in the two cells that you started with now into 4 cells. So at the end of telophase 2, you have 4 haploid cells, each of which contains a single chromosome. There's no pairs of anything, no pairs of homologous chromosomes, no pairs of sister chromatids, everything single and on its own. I want to remind you that the entire purpose of performing meiosis is to produce gametes, to produce sex cells. So now we're going to talk briefly about the process of making those gametes. Don't get too excited, kiddos. When we're talking about the process of making gametes, of course, it being biology, we're going to introduce a bunch of gigantor words. So, gametogenesis is the process of making gametes. Well, you should have learned by now what a gamete is. Remember, gametes meet and then they make a baby? Genesis. Any of you guys who are familiar with terms in the Bible, what is Genesis? Well, Genesis is the beginning. That's what Genesis literally means, the birth of, the making of, the beginning of. So gametogenesis literally means making gametes. It's almost like saying gametosynthesis, if you wanted. So we have two processes that make gametes, because we have two different types of gametes. Spermatogenesis makes sperm. Should be pretty easy to remember. It's got the word sperm in it. And it produces 4 gametes, 4 sperm of equal size. Oogenesis, on the other hand, is what makes eggs. I know some Latin roots. I know that ova means egg, so that's a little easier for me to remember. What you might want to look at is the fact that those O's in the word oogenesis look kind of like eggs to help you remember that that's what it's making. Oogenesis, on the other hand, makes only one really big gamete, and we're going to look at some images of that on the next slide. So, there is a lot of stuff going on in this diagram, but first I want to concentrate on the left-hand side with oogenesis, or making eggs. What you'll see is you start out with a cell that has 2N in the middle. What that means is it's diploid, it's got 2 sets of chromosomes. After the first division that you see there, you'll see that the large cell that's left behind now only has 1 N in it. That's because it's now a haploid cell. You also see something that's labeled first polar body. The name of it's not important. What's important to note is it's not much more than a little bit of DNA. That's basically junk DNA that gets thrown out. Then, right before fertilization happens, you have a second what they call polar body coming out, and that's going to remove basically the sister chromatids that come out at the end of meiosis 2. So you have one very large cell left, and the divisions were mainly only to get rid of DNA. On the other hand, if you look at spermatogenesis on the right-hand side, you start out with 2N in that cell where it says primary spermatocyte. Once again, the words aren't important. You just look at it and see that, okay, we divide now into 2 cells, each of which is haploid. It's got 1 N, 1 set of chromosomes. Each of those divides into 2 cells, and you end up with 4 cells, 4 sperm of equal size. Finally, at the very end, you see a sperm fertilizing an egg cell. You get that fertilized egg cell, which is, as we learned earlier, called a zygote. And that zygote is now 2N, or it's diploid. It's got 2 sets of chromosomes. And in the case of humans, that means 46 chromosomes. Now we come to the end of our lesson where I just want to contrast and compare a few things between mitosis and meiosis. So mitosis is what produces somatic cells. These are body cells. Most of the cells in your body are somatic. And these cells that are produced are diploid and they are exact clones. So they're exactly genetically like the mother cell. Meiosis, on the other hand, produces gametes, which means sex cells, sperm, and egg cells. These guys are haploid. They have half the DNA of normal body cells. They are genetically different from the original cell because they only have half of the genes, or half of the copies of genes that you have in a normal body cell. I hope this lesson has been at least somewhat helpful for you, and if it hasn't really helped you out much, don't worry, we'll be going over this in class. Have a good day, evening, or whatever time period it is that you're watching this.